In this talk, we're going to learn about proteins, which are macromolecules found in living systems. We will first begin with the organization of multicellular organisms and the different macromolecules that are found in cells. We will then learn more about what proteins are and what they're made of, along with the bonds that attach the different monomeric units that will form proteins. We will also learn about protein folding and describe the different functions of proteins in a cell. A multicellular organism like a human is made up of different organ systems. These organ systems include the digestive system, respiratory system, excretory system, so on and so forth. The organ systems like the digestive system shown here are in turn made up of organs. These organs can be, for example, the stomach of the digestive system or the liver, and so on and so forth. Organs, in turn, are made up of tissues. Tissues are made up of cells, and when we look at cells of multicellular organisms, they tend to be eukaryotic in nature, and hence they have organelles. Additionally, cells and these organelles are made up of macromolecules. Macromolecules are larger molecules, and hence we can say in a certain regard that they are made up of molecules, which are in turn made up of atoms. Living cells are made up of 4 different types of macromolecules. In many instances, these macromolecules are polymeric in nature, in that they are made up of repeating units that are linked together. Of the 4 main types of macromolecules that we find in biological systems, their names are carbohydrates, proteins, lipids, and nucleic acids. In this talk, we are going to focus on proteins. Proteins can also be called as polypeptides. However, it should be noted that there is a subtle difference between proteins and polypeptides. Polypeptides are polymers of amino acids, and hence the monomeric unit of a polypeptide is a molecule called as an amino acid. Amino acids bear that name because they have an NH2, which is also called as an amino group, and they also have a COOH group, which is called as a carboxylic acid group. And hence these molecules have both the amino group and carboxylic group, giving the name amino acid. In addition to these groups, we can see that they also have a hydrogen that is attached to that alpha carbon. And hence when we look at the box, the arrangement of atoms that we see in the amino acid, this specific set is going to remain constant in the different amino acids that are found in the body. The part of the structure which is shown by the side chain group is the part that shows variation. Now, in our body, there are 20 different amino acids, which means that R part of the amino acid is different in each of these 20 amino acids. Here we can see the structures of the 20 amino acids that are found in polypeptides. Based on the R group, they can be assigned into different groups like aliphatic amino acids, aromatic amino acids, so on and so forth. Every amino acid can be denoted by a 3-letter abbreviation that is shown in the blue boxes. Additionally, every amino acid has a single letter code that can also be used to represent its name. In this table, we can see all the 20 amino acids assigned into different groups based on their side chains, along with their 3-letter codes and 1-letter code. 2 amino acids can be linked together, and for this, they use the amino group. And in this figure, we are able to see the amino group of one amino acid can react with the carboxylic group of another amino acid. And when this reaction occurs, water is lost, and the 2 amino acids they can be linked together through a bond that is called as a peptide bond. The peptide bond is essentially an amide, but in the context of amino acids, it is also given the name of a peptide bond. And this way, we are able to see two amino acids being linked together to form a dipeptide. When up to 20 amino acids are joined together, the molecule that is formed is called as an oligopeptide. Now, in an oligopeptide, when we look at one of the ends, there's always going to be a free amino group, and this part is called as the N-terminus or amino terminus. When we look at the other end of the chain, we are able to see a free COOH or a COO minus group, which is the carboxylic acid terminus or the C terminus. Thus, in oligopeptides as well as in polypeptides, we observe an amino terminus end and a carboxyl terminus end. In between, we are able to see the peptide bonds that are linking the amino acids together in a covalent manner. When we have more than 20 amino acids covalently joined together, then we are able to obtain a polypeptide. The polypeptide will also have an amino terminus, but there is a free amino group, and a carboxyl terminus, or the C terminus, where we have a free carboxylic acid group. Polypeptides are made when these amino acids are joined together in a linear manner to form a linear chain. However, this linear chain does not remain open as shown in the top part of the figure, which shows unfolded polypeptide. They will fold to form three-dimensional structures. This three-dimensional structure is also called as the conformation of the polypeptide. And the conformation or the three-dimensional structure is determined by non-covalent interactions between the different R groups or side chains of amino acids. So in the three-dimensional structure that is shown here, we are able to see right in the center or in the core, the hydrophobic amino acids are interacting with one another through their side chains, while towards the outer side of the polypeptide, we can see hydrophilic side chains of amino acids that are interacting with the outside of the polypeptide. There are different levels of folding when it comes to polypeptides. The first level of folding is the primary structure, and this is where we have the linear chain of amino acids, and hence we know the amino acid sequence of the polypeptide. The amino acids can start folding to form more complex structures, and these are in the form of secondary structures. Two very common secondary structures is an alpha helix, which is a helical structure that is made through the arrangement of the amino acids, and we also have ribbon-like structures called as beta sheets. And thus, these secondary structures can further fold to form a three-dimensional tertiary structure that we can see here. Now, in some cases, you can have a quaternary structure where you can have multiple polypeptides associating with one another, and such structures are called as proteins. And thus, a protein can be made up of many different polypeptides that associate with one another. When we look at the function of proteins or polypeptides, we can consider them as the workers of the cell, and hence they have many different functions. One example of a function of a polypeptide is catalysis. Catalysis is a process where a molecule can help in a chemical reaction to proceed faster. In such scenarios, the enzymes, which are the proteins that mediate catalysis, can interact with a reactant or a substrate and help to form a product. It's able to do that by binding to the substrate and mediating the reaction to go faster, and once the products are formed, they dissociate from the protein or the enzyme. Another function of polypeptides is to mediate transport. So in this figure, we are able to see two different motor proteins, the kinesins and the dyneins. And we're able to see how they're able to bind to cargo, which in this case is a vesicle, and transport that cargo from one part of the cell to another part of the cell. Another function that proteins have is to regulate the structure of a cell and in certain cases aid in movement. This is done by a class of proteins called as cytoskeletal proteins, and three common cytoskeletal proteins are microtubules, microfilaments, and intermediate filaments. Another function of polypeptides is to mediate the transport of molecules from the outside of the cell to the inside of the cell or vice versa. Another important function of polypeptide is to help a cell to sense what is present in the environment and then respond to it. For example, here we can see a protein that is present on the surface of the cell, and it has the ability to bind the molecule insulin, which is a hormone. When insulin is able to bind to this protein, the cell on which this protein resides is able to know that there is glucose in the environment, and hence it will start the process of transporting that glucose inside the cell so that it can use it to generate energy. With this, we come to the end of our talk, where we learned about the organization of multicellular organisms and the different macromolecules that make up the cells of the multicellular organisms. We also learned about proteins that are made up of amino acids through the formation of peptide bonds. We then looked at different levels of protein folding and some of the functions of proteins.